How do I do that? Along with Bryson.
Okay. So people just missed my intro and my acknowledgement of our partners on audio. So because I'm part of the Reef team and a very proud member of the Reef team, I just wanted to spend a minute to talk about myself, but to just tell you guys a little bit about myself. So I reside in Orlando, and I do come down, especially in the warmer months right now, about every other month to do the smile dives. Yes, I do go to Disney a lot, and I have 2 kids and my husband that are not watching because they're out doing all their kid activities for the evening. But this all started for me since I was their age, younger than their age. I wanted to be a marine biologist like most kids do at some point in their life. I just hung on to that a little too hard. So this has been a lifelong pursuit. I went to FSU for my bachelor's degree, so I'm also a diehard Seminole. I will not acknowledge, sorry, those other schools, but I am a Seminole through and through. So I'm just, luckily no one's walked out on me. That's good. But during that time, I also interned at Moat Marine Lab. And so I was actually interested in sharks initially, like also probably a lot of people that are interested in the field. But during my work with Moat is when I started working with snook. And that kind of led me down this path of bony fishes and doing fisheries ecology, and have never looked back. So I blame them. I did my master's degree through Northeastern's 3 Cs program. So I was in 3 different locations in Boston. That's a picture of me in Moorea, French Polynesia, and Catalina Island, California. But my actual thesis work was performed in Moorea. After my master's, while I was waiting out an economic recession to try and start my PhD program, because we are like funded for those programs, I did some other jobs where I did a stint on land as my joke. I worked for a local county government in Orlando doing land conservation and acquisition. So I worked with wetlands and upland systems. So I was into birds and stuff for a hot minute there, but my calling was always back with the fish. I also worked at Disney's Epcot Aquarium. So I have also been in the fish bowl myself and have a feeling for what that's like. And then I've taught college biology for over 10 years throughout all of this kind of stuff in the background, as well as during my PhD. So I did that at UCF. And I worked with sport fishes, so like sea trout, snook, snapper, red drum, that kind of thing, with oyster reef and living shoreline restoration. So all of my background is in fish, like in just different facets of fish and their relationships to habitats, and also with people, because I work with a lot with fishing targeted fishes. So now I have actually, because of this postdoc research position that I have with Reef, I can now say that I've performed research on all 3 coasts of Florida. So I am not only a Floridian, but I am very immersed in fish diversity in each of these sections. So you guys can still completely out ID me on the reefs out here. But I'm pretty strong on oyster reefs and seagrass. So I'm not perfect, but got it kind of all over the place. So enough about me and more for the project and why we're actually here, which is to talk about some of the needs that fisheries stock assessments require. So a lot of the things that go into fishery stock assessments require different biometrics, so different information about the fish and their population. A big, huge facet of that is size data, specifically length. And so I'll talk more about that in a second, but it lets us know a kind of a snapshot about the population and how that can relate to fishing pressure and environmental change. So size is a really big cue. Traditionally, lengths are obtained through harvesting a fish. So we get that information from the commercial fishery. When they're bringing in their catch, they can get measured, or they're reporting those measurements. Or if you're a recreational angler, you may have been asked at boat ramps or docks before to provide information to FWC, for example, about the length of your catch. Or they'll just take it from you and measure it for you. Other methods are handling fish. So this is me in the field during my dissertation work with a ruler that's far too small for that snook, but we made it work. So that's usually the length of fish I was working with. But even that, like fish that can be released, it still causes stress to that individual when they're being handled and taken out of the water and measured. And of course, when they're harvested, they're lost from the population. So those are some risks, I guess, to doing these direct traditional methods, but there's also a limited distribution of sizes and species when we do that. especially with the fishing industry, because they're restricted to how big of of species they can bring in, what species they're allowed to bring in. So we really don't know a whole lot about the general population of these fishes. So there are other existing in situ methods, which means those that are done directly in the field. And in this case, I'm talking about ones that don't involve contact with the fish. So one such method is done by the federal and state agencies. It's called the RVC method, the Reef Visual Census. So these are professional scientists that are trained just like Reef does with like fish ID and doing estimates of abundance. And they do this visually underwater in like a very defined space and time. They are also trained to estimate fish length from a distance. So that's what this T-bar is. It's a very fancy PVC pole with a ruler on it. So they use that to assist, but they're very heavily trained. I think they have to get like tested every year to make sure that they're up to par to be able to do this. So it's a very resource intensive method. And not everybody can do it, and it's pretty tedious too. Other methods that are used are ones that if you've dove with me before, you've seen this equipment. This is a stereo video camera, which is like a dual GoPro system, and it basically works like how your vision does to kind of triangulate the size of a fish. It does so using this big, bulky, expensive system, and then it does so in post-processing. So when I'm home in Orlando, this is a lot of what I'm doing right now is completing the post-processing of those videos. So it also requires software that's several $1,000s, and it creates it's kind of tedious work too. But AI might build that up for us over time. But still not really conducive to doing a reef survey. No way could you guys do a reef survey with that big, bulky camera system. There's other methods like laser calipers. So you have a known distance apart that those two lasers are. So you get them on the side of a fish and you can estimate length in post-image processing as well. So these are some of the methods that we currently use, but not all of them are really conducive to doing with recreational divers who are out on the reefs all the time. But back to why we even care about fish length. As I mentioned, fishery stock assessments incorporate fish data. It's a major biometric that we use. Here's one such display of that. It's called a length frequency distribution. So on this x-axis on the bottom would be different bins of length. So this is fork length in millimeters. On the y-axis is how many individuals in that bin of length. Basically, you've got a nice bell curve, okay? If I measured everybody's heights in this room, we would probably have some sort of bell curve where there's fewer of us that are short, fewer of us that are tall. Most individuals would probably be in the average height range. So animal populations seem to fit this kind of criteria, but it can also be informative if you have much more individuals in the smaller ranges. That might tell you that your population has had a recent strong recruitment class, show some growth, or you can have decline, so your population wouldn't be getting to the ages that we want to see. Some of these are kind of funky when you have slot limits for recreational species. There might be like a gap where it's very heavy fishing pressure, and then you see the sizes on each side. So it's actually really reflective of what the population is experiencing. And we can even look at these through time to try and kind of pinpoint when those environmental perturbations take place or when you have different fishing regulations at play. The other thing, so it's coming out a little blurry, but you can hopefully get the idea with these curves. They also reflect the age structure of a population because we can correlate height to age with fishes. So this is done, with a lot of species, with probably most common species or species of interest, is that you can measure your fish. This is one that would be humanely harvested, and you extract the ear bones from fish. Does anybody know what we do with the ear bones on fish? Otoliths. Yeah. Exactly. So otoliths are these little ear bones inside the fish. They lay down a ring every year. When they're young, they lay down daily rings too. And that you can actually age an otolith to count the rings just like on a tree to tell how old a fish is. So when you've got a harvested fish, or just a scientifically harvested one like I did with my dissertation, now I get to keep everything alive. It's great. So I really like these methods that keeps everything alive. But you can tell how old the fish is and how it relates to their size. So that when we are measuring fish through these non-lethal methods, we can take a look at what their estimated length is. I think it's flipped on this curve. So their length that we're measuring, and you can extract out their estimated age from there. So you're also getting an idea of how old the population is. So we have age and length relationships, but there's also a relationship to reproductive potential. So we know that the older, larger fish tend to have higher fecundity or a greater potential to carry on the next generation. And that is really, again, related to size, but it's also important for fisheries regulations so that we can keep the populations going based on how we set these different limits. It's also related to mass. So there would be curves like this with length and mass too. So you can also estimate biomass of the population as well. So all of this information helps set our fisheries regulations, and that's why regulations change, because the population may change. Maybe we went too far with a regulation and we need to restrict it. And there's always, I'm sure you hear about fishery regulations changing all the time, and they're usually pretty controversial. But they try to get the best data they can to make these regulations. And then they open it to public comment and, et cetera. But all this actually will feed back into your length estimations as well. So it's kind of a cycle with what we do with this data and why it matters. Now, I don't really think I need to tell much about this project here, but does anybody in here conduct surveys for Reef? Yeah, few of you. And obviously you all know very well about Reef, but this is probably what we are best known for, is our volunteer fish survey project. I think it's pretty synonymous with Reef. This involves roving divers that have been trained in fish ID, and you can start at any level, of course, many of you know that, and you can work your way up to more expert levels, gaining your fish ID knowledge. So the divers would go out with a dive slate with fish species from their region. They would estimate relative abundance of each species that they encounter. So you would have options with single, few, many, or abundant for each of those species that you encounter. You can even do like a pretend survey from the fish on these walls around you. Like Sergeant Major would probably be abundant, whereas NASA grouper behind me might be a single experience. So they have that for every region, and we have a membership of over 85,000 incredible members for Reef that have amassed over 300,000 Reef surveys worldwide over the last 31 years. Is that where we're at? And this matters. It is directly used in scientific publications because this is publicly available data. So the database can report sighting frequency and density and things of that nature for the fish that are encountered in all these different regions around the world. And it can also report like novice versus expert surveys. So there's different ways that you can filter it. You can filter reports by region, and then we can also people can contact Christy, our co-director, if they want more expansive data sets. So they've contributed to over 130 scientific papers. And even so, recreational divers are still an underutilized source of data collection. So the South Atlantic Fisheries Management Council, who I mentioned is probably our biggest partner in terms of putting this grant together, and I'll be presenting to them next month or next week. Jeez. Gotta get ready for that. I'm good. I'm just kidding. But they have never worked with divers ever for data. They only have like 50 black grouper lengths in their data set. So this is a huge opportunity for them. And we will continue to show that proof to them that why it's working and why they should continue to consider divers as a source of data because it can fill these valuable data gaps in fisheries. So that brings us specifically to the SMILE project and how it can fill those needs. So again, Size Matters Innovative Length Estimates. It is designed to collect fish length measurements with an in situ tool used by citizen scientists. So we have our nice, easy to use camera, much better than the bulky stereo video system. We have one cool laser. It's like playing laser tag with fish on the reef. And its goal is to complement the existing reef fish surveys, because now we can task divers to do one more thing with us, which is collect fish lengths, but not on everything, just 11 species. And that list could be, modified over time. And it's with the idea that it's going to improve future stock assessments for data limited species like black grouper that we don't have a lot of information on, and ultimately inform management and conservation efforts for that population, for that species. So this is really a project that's really strongly fisheries ecology and very strongly citizen science and conservation technology and AI. So we're kind of bringing all these things together, and you guys can be on the cutting edge of this brand new tool as we continue to implement it. So the way that this works is where we're collecting data for this so far has been focused here in the wonderful Florida Keys, mostly at Isla Morada, which I'll talk about in a minute, but we still do some Key Largo dives. And I'll come back to this, but you guys can also take the cameras out wherever you want. But when you're diving with me, these are where we're going. So the camera, it's abbreviated Fish Sense Light, but it is used by citizen science divers to collect images with a laser on the lateral or broadside of a fish for 11 target species. I will often go out with a stereo video camera. So if you've dove with me, you have seen that unit. So I am the staff in that sentence. But all of our fellows and fellow Reef employees have also taken out that system. And I use it as a method of comparison. So we have a known camera system, and now we're bringing in a new one. So how do those two systems relate? How good are they at matching up? This is a highly collaborative project, as I mentioned, but we also have, in addition to all these partners that we meet with once a month, the engineers we meet with like every other week, we have a stakeholder panel that's made up of government agencies, private industries, and we have Fred as our citizen scientist representative. We put Fred to work on this project in every angle. So thanks, Fred. And this also gives us direct input on how we want to design the formal tests, how the cameras are performing from citizen scientists. It's not just Fred, but whoever is coming out with me also gives me that input. But we also have the end data users. So that's kind of the term for anybody that's going to use data. It's kind of one of those like newer, frequently used terms. Is there our end users. So how are the government agencies going to use it and what's ideal to them? So we've been focusing our efforts on alligator reef. And the reason why that is because we have 11 target species that, don't worry, I will show you who they are in just a few minutes. But I went into the reef database for this. So we identified our species first with our collaborators. We had a list of reefs on the table that we were interested in looking at, and I went into the reef database and saw where we had most of them to be able to encounter them and put this method to the test. The other cool thing about working with alligator is that we could have done this in some of the Key Largo reefs, which is why they were considered as well, but they offer this other ecological opportunity and that they are a sanctuary preservation area. And so with alligator, it's currently in this red polygon is the spa, the sanctuary preservation area. We can sample inside and outside of that spa. And the proposal, though I know that it hasn't gone well recently, so hopefully that's not the end of the story, but it was proposed to be extended to the, was it the 90-foot contour or the 60? Now I'm blanking. I think it was the 90-foot contour because that's where we'll dive down to. So we're able to sample outside of the spa with the idea that if there's future protection in that area, we have a snapshot of what's happened before and after the protection status. So I can tell you without quantifying it, there's an absolute difference in these two areas. So I look forward to quantifying it and showing that evidence. So our handy tool here is the SMILE camera, or the Fish Sense Light, as it's also known as. But we usually, within Reef, we call it the SMILE camera. So it is an Olympus TG6 camera. So that was a tried and true camera that's been used, and it's also used frequently by recreational divers. So we know that it kind of has like, people are experienced with it and it's easy to use. It's got its typical underwater housing, and then we have a detachable wide angle lens that's on this. We're hoping to reduce the need for this in the future, but right now, to answer any questions, we use it to reduce distortion on the edges of the images. So right now it's an important component, but they're trying to look to remove that. And then in the mount, we have a cold shoe mount for this laser, which now has a nice metal, robust mount that doesn't, I don't want to jinx it, but it holds up really well. And we have a single laser, and the relationship of this laser position is known to our engineering team, the engineers for exploration at UC San Diego. They also calibrate the camera to the housing, to the lens. So this whole unit is a known entity to them when they're doing the post-processing of these images. And then we also partner with Backscatter to get this. Backscatter, I don't actually have a slide for this talk, but we have a little bit of laser issues. So anybody that's dove with me over the last couple of years, we've been through lasers like no other. They flood, so it's our problem. So we're making our own laser. So we've had enough of that. And we might even it might be a dual laser coming soon. So stay tuned. But we have laser prototypes in the works. Allie's tested them out on Grouper Moon in the Caymans. So I'm not going to go through all this, but just so you know, when you're using the camera, there's just certain settings that have to be applied. This is all boring stuff, but for people that are into photography, just know like you can't just kind of set it however you want. It does have certain criteria, such as no flash, because that creates backscatter. Burst mode, because we want several images of the same fish. We know we're not going to be perfect in taking the length of that fish, and we also can't prove that it's that length, right? I can't go and reverse these methods and grab the fish and say that's how big it is. But the more pictures that we have of it, we can build like a mean and some variance around the size of it. And then we also have to do the raw image file because that's part of their computing process for the length. For taking them out on the water, these are things I go through. So like if you're going to take the cameras out, I have a webinar available that goes through this in more detail. And then when you're picking up the camera to use from Reef, or I'm meeting you dockside, I go over these items again and let people kind of get a feel for the camera. But it's really that easy to use that I can just do this virtually and kind of walk you through camera use. So it's generally just checking those camera settings, and usually Reef staff has done that. Once you go in the water, you have to burp the lens, which is a fun word for just loosening it and tightening it again, because you want to flood water between the housing and the lens. Turn on the laser, and then we have currently a preset dive slate that we take pictures of to help calibrate the system. Basically, that tells us if the laser is moving out of its expected range or not. But as we move forward with our own laser prototype, we're hoping to remove the need for the calibration slate as well. So stay tuned. It's a tech project. It's ever evolving. And then you guys get to just perform your dive like normal. So you can do a reef survey while you're doing this. And when you encounter these 11 species, you're taking pictures of them with the laser on the side of them and doing your burst mode to get the images of it. We have, different best practices that I go through with people, which is you want it as broadside to the fish as possible. You want to be optimally like 3 meters away, but it can work within a 0.5 meter to 5 meters. Probably depends on your visibility, probably depends on the fish's behavior. So there's a lot of kind of factors in there, but it's also so easy that if like the laser is on a fish, I forget I have fish in here. Oh, there's no battery in this one. Awesome. They're all literally splayed out on the counter from taking them out in the field today. But you would just once the laser dot is on the side of a fish, you don't even have to look through the camera. You just push the trigger and it's going to be on it. So it's literally set that you never even have to look through the camera. I got you guys all excited about a laser and let you down. And then we just ask people, like, don't come back and take a picture of the same Goliath grouper. If you do, tell us that you're doing it later on, reporting it, so we don't report that there's 2 Goliath grouper of the same size. So there's just little things like that to be cognizant of. But speaking of, here is our curated list of reef fish. This has been carefully curated based on their economic and ecological importance, and especially those that are data limited, that are fished, like the groupers and snappers and hogfish. And then we have 4 species of parrotfish that give us an idea of like an ecological indicator on the reef, since they are herbivores and occasional corallivores. So that gives us especially for the Florida Keys, an idea. So a lot of you guys are familiar with these species, but they also have little behavioral quirks when it comes to photographing them, which is kind of funny. So the black grouper plays a game of cat and mouse with us. So the black grouper wants to be photographed, but doesn't want to be photographed, or it wants just the right angle, swims away, it comes back. They're very funny. You can ultimately get them, but they're not going to make it easy on you. Nassau grouper are pretty chill for the most part. So Nassau's have that nice Y shape on their forehead, and we all saw Nassau today, which in Key Largo, which is pretty cool. And they're pretty chill about being photographed. They have the bars on them, those nice red bars, and are really indicative by that black patch on their caudal peduncle or their saddle. And then there's red grouper that I so wish we had more red grouper. If you have a site where you have lots of red grouper, please take a smile camera. They are the easiest thing to photograph. You could hug a red grouper if you wanted to. And that's probably why they get speared so easily. I know, it makes me very sad. They're a very trusting fish. And then you have Goliath grouper, which are like the monsters on the reef. They're pretty hit or miss too. They're pretty chill for the most part, as any diver knows from encountering them. But on occasion, they're gonna swim away and make it hard. Hogfish are another big species that's especially important to the Fishery Management Council. They have, you know, these kind of like flanges to their dorsal fin, but they can be that coloration with like the patch on their forehead. They could be really flushed out, especially over the sand, or they can have that like mottled appearance. They're just a really entertaining fish. Some of them don't like the laser on them, and others don't care. Others chase the laser. So there's a lot of that. There's like, have you ever seen a cat with a laser pointer? Enjoy the grunts and the yellowtails. There's like certain things that really want to eat the laser, but so you can have other kinds of fun with these cameras too. Then there's the gray or mangrove snapper. They're kind of drab on the reef, but more colorful when they're in the mangroves, but they will often be schooling. There was a lot of those today, actually. Mutton snapper are pretty curious, so they'll come around. They don't really tend to mind being photographed. They have these faint white bars and then that indicative like black spot on its dorsal lateral line. And their face is pretty smooth. And they're usually solitary, or they might have another one hanging around in my experience. But they're kind of curious. They'll just linger nearby. And then we have our 4 parrotfish species. I bet you guys never encounter stoplight parrotfish, right? Super rare, these stoplights. That's actually kind of part of their selection too, is like, we know you'll always going to find stoplight parrotfish on the reef, and they come in these nice 3 different sizes and color patterns, where you have the juvenile phase that has kind of these little white specks, but look kind of like an intermediate with these white specks. The intermediate phase looks like a pine cone to me because its scales are so outlined. The terminal phase of the stoplight has a yellow right at its operculum, and then a yellow blotch on its caudal peduncle as well. They're very zoomy on the reef. Have you guys ever noticed that? When you're trying to measure fish on a stereo video camera with 2 GoPros, you really notice how zoomy stoplight parrotfish are. So they are just all over the place. And then parrotfish just always do their thing, so you just kind of have to get creative. Rainbows are the orange and green big parrotfish. Midnights, and they kind of have that typical parrotfish beak. Midnights are a deep blue with this like light cerulean blue patches. They're really pretty. None of them seem to care about being photographed. And then blue parrotfish are a nice powder blue. The young sometimes have like a yellow, sometimes like I've seen like an orangey pink patch on them, but a really blunt forehead. So even in video, when I don't have it clear, I can see their silhouette that it's a blue parrotfish. A lot of these you can figure out from a silhouette too, but that really sets the blue parrotfish apart. So these are our current 11 species. Again, that could modify based on fishery management needs in the years to come, but this is where we're at right now. So just a couple of examples of what we're looking for, what we experience. This is a really high quality example from the Caymans. So this is not going to look like this. Okay, it's going to look more like this. I think this is like Tom's video too of a smile camera doing its thing. But this is kind of what we're looking for. If it's not in the center, that's okay. They can still work with it. If the fish is slightly angled to or away from you, that's okay. They can work with it. But ideally, we're getting as on the side as we can. These are in test dives from California. We lose red lasers on dark fish. So there actually is a laser on that midnight, but it's very hard to see. Some of the poor examples, and this isn't anybody being like bad at taking a picture. You're dealing with a moving animal and you're trying to put a laser on it. So sometimes you're doing a burst mode. So you're just grabbing pictures. But these are ones that we're not looking for, but they're in the data set and we just don't use them. It's not a big deal. But like what that angle would be too harsh. This one would be too harsh to try and grab a size on. These are from California too. This like isn't even on a fish. This one wouldn't even be centered. That might not even be on a fish either. So there's definitely like good, bad, and ugly for the photos, but I don't want anyone, it's like sometimes people feel intimidated to start the project because they're afraid to, I think it was Laura Lee who used to be scared because they're afraid of giving me bad data. And we're like, no, that's actually what's so great and convincing about this project is we're not asking a citizen scientist to go and lay a ruler on something and have to try and think they need to be precise. You're taking the images and then we work with it after the fact. So it's on us to get the measurement right. You just have to take the pictures. So a little bit about how that works. So our engineers work with the calculations of this. And I do my best to put that into layman's terms. I need them to put it in layman's terms to me. So the way that these cameras work, because there's usually a lot of curiosity for it, we're pretending these numbers are lined up. But basically, this would be like a coordinate system. So you have values associated to every position in this photo. And it relies on depth of field. So things that are farther away appear smaller. Things that are closer up appear larger. That's why we need the laser, for it to tell us how far away that thing actually is. so that we can accurately estimate its size. And so if we were to put a laser on this fish right here, that's what its coordinates would be. It would have an XYZ position in that photo that allows them to tell them where it is in the photo, and it's used as a scaler that I can kind of show you in a second to estimate the length. If that fish moves closer, my laser position changes. So here it was further away, here it is closer up. So now its XYZ coordinates are in a different position. So that allows them to find the laser in the image. They then need to identify the head and the tail of this fish. So they would be able to mark that in the image. And then this is where the raw image file comes into play. So we're actually hoping, we keep pushing to do this on video down the road with GoPros, but we're kind of locked into the raw image file right now. They have faith that it could be done, but this is where we are and why. So it relies on the pixels. How many pixels that fish takes up, like end to end in the photo in relation to where the laser position is, tells them that's the scalar within that image, and they can then calculate the length of the fish itself. So it's pretty ingenious. And because of all these other methods that I've told you about, even other scientists in the field are like, how are you doing this with one laser? Well, I'm not, but I can tell you how they're doing it. And so it's a pretty effective method. And there's so many bars on this picture, but you don't have to worry about all of it. These are all individual fish that were, like, I followed somebody with a smile camera with a stereo video camera. So I matched up exact fish on these two systems and put them side by side. So these are all just individuals on the x-axis. The y is mean fork length. The blue is the stereo video dual GoPro camera system. The tan is the fish sense light. So they're pretty even when I do statistical analyses on this group of fish, and they're not statistically different from each other. So they're estimating length about the same. There's obvious outliers to that, and the stereo video system does actually tend to measure a little bit larger, or depending on your outlook on life, the fish sense measures a little smaller. I don't know which way you want to look at it. But that's kind of the trend that I see with it. But again, they're just like statistically, they're not different. So we've also taken out fake fish in the pool and in the field so that we have something that we know what its size is. We put both camera systems up against it to see how they shake out. And this, that's me holding a fake snook. I think this image is from a fake shark, but anyway, like it's actually the reject fish from the wall, the giving wall out there are some of my fake fish. So, but I like to use my realistic looking fish. I digress. So the dotted line here is the actual size of the fake fish. And then the bar graphs tell you the length that these camera systems were measuring. So we're still building out all these systems, and we're doing that with the help of citizen scientists too. So we can strengthen this with different fish models and more than one smile camera. Because in science, we like replicates. We have to prove that it's not just one camera or one fish model that it's able to do it on. We have to show that it can do it across the board. So that's the science behind it. Now the citizen science participation is the crucial part because you guys are the people that are taking the images. So far, I have to update these numbers after this week, but we've had 46 different volunteers, and many of you are repeat divers for me, that have done 295 total dives in the Keys with the smile cameras, and it's now been on eight reef survey trips. I should have updated this actually before this talk. It just went last week. And we are working on developing partnerships with local dive operators to house cameras at a dive operator so that they can do a simplified webinar that's not as long as my rambling here, to get people up to speed on using the camera with quick training and just learning 11 species. So it's not as intimidating as 300 species in the tropical Western Atlantic that we have for reef survey potential, but just 11 on our list. And then we have developed a formal survey to examine motivators and barriers to participation in projects like this, and also give us ideas for an idea of how people are enjoying the cameras, how they're performing. So we also have very specific questions about the smile cameras themselves. So one of my incredible groups last, I think August at Alligator. So the work for this project has just begun. We still need help for it. And so I'll go through the different opportunities that we still have. But don't we look like we're having fun? I think these are all taken after diving, and we're still smiling. Get it? I know. I know. Sorry. Can't help myself. So some of the dive opportunities that we have, we've hosted workshops. I don't know that we have any more in the current grant world, but we've applied for more money, so we might be able to keep doing these workshops where Reef staff here in the Keys will host an in-person workshop like this, and then we'll go and do dives on the reef and get like literally pulling people out there and getting them to use it. There's the opportunistic use with local dive operators or here at Reef. If you want to check out a Smile camera, we'll set you up with all the goods and make sure you're trained. And you're giving us the information of where you dove and when and with what camera. Those are kind of the basics that I need, is where it was, when, and what time, that kind of stuff. And go out and collect data. And right now we'll be downloading it. Down the road, we're also trying to scale this project up. So not only using it here in the Upper Keys, but get it across the Florida Keys and build out a website where people can download their own data and then upload, sorry. And then Fred always wants to see his photos. This would also be an opportunity for people to see their photos as opposed to never getting them back from me because I'm dealing with all the other stuff with it. So there's ideas that we have and paths forward on how to do that and hoping to get more funding to support that. Other ways that we have been involving citizen scientists is in the back end. So doing all of this image processing with our engineering team is hopefully an automated process down the road. But with all machine learning, we have to get there. And so it's kind of manual at the moment. So we've had 60,000 images annotated for laser position. We set people up remotely, so I never have to be in person with you to do this. And John hosted an annotation party last week, both in person and online, to help support this as well. So we do items where you identify the laser position, you identify head and tail, which is the project we're on right now. So that, again, eventually AI can start finding, okay, here's what a laser looks like. Do you know how many green glowy things there are underwater? Because we've switched to a green laser. Never noticed how many green glowy things there are until you go to annotate and look for a laser in that picture. So we need to make sure AI knows what it's looking for and the shape, especially our reef surveyors. We're going to ask that you guys start moving on to fish ID for us once that is ready to go. Basically, you're going to find the fish that the laser is on and you'll say that's a mangrove snapper. So it can start doing that for us. So we don't have to do all this manual labor. See if, I think this one links externally. That's okay. But it just shows you how to do the laser. It used to be embedded, I promise. So then the other item that we have right now is our citizen science survey. So we have been building out a survey for the last year with our partners at Colorado State and FWC. So there are social scientists that structure surveys, how questions are, I don't even think you ever knew, because I didn't, how surveys are made. But there is a science literally to it with social science and how questions are structured in order and what we're trying to get at. So we have finally completed the development of that survey. This is Reef's first ever formal survey. So this is a really cool opportunity in Reef's 30 years of doing the Reef, volunteer fish survey project. We've never surveyed the humans in a formal way. I'm sure you've been asked things after Reef Fest and the Derby and things like that. You've given us feedback, but we've never done a scientific survey. So we have access to that through the QR code, and it's also on our website. But the idea for this is to examine motivators and barriers for participation in SMILE. So it gives you a little background on SMILE, but you're all experts now, so you're good. But it also asks about conservation technology more broadly. Here's an example of things. What motivates you to participate in marine species monitoring, such as reef surveys or the SMILE project? What level of interest would you have in using a camera as part of the project? It'll ask about dive experience, camera usage. What do you normally use when you're diving? And if you've already done a SMILE survey and a survey, how easy was it to perform both? What type of training would you prefer? So it's got specific questions for us to keep improving the project itself and, again, scaling it up, but also informing conservation technology more broadly. So all of our partners on this are really excited about this survey, which is probably why we have such great support for it, because they're all really interested in what you guys have to say. So come say it in our survey. And then just again, I'm going to sell it to you one more time. Why should you participate in SMILE? It helps scientists and fisheries managers fill these important data gaps. They need the data, especially we are very resource limited in the field. And so we can assist with that. And that's why citizen science is such a growing field. You can get involved with new science. Like I said, you are on the cutting edge of this new product, this new technology that we're using. It's fun and easy. Who doesn't want to play laser tag with fish on a reef? It gives you a new challenge. If you're not great at fish ID, but you can handle a dozen species and you want to contribute data still, this is a great opportunity for you. So with that, I will wrap it up. This is our, you guys could have all just gotten this out of an infographic. You know, I'm just kidding. So this like summarizes, this summarizes really what SMILE is about. It's a flyer that we've had made, how SMILE addresses some of these things, and our QR code again to get more involved, because obviously we have lots going on with this project. So I'm happy to take any questions that you guys have for me, and thank you again for your time and your support. Yep.
Oh, sorry. Well, actually, I have two. One is more about the science of it.
Mm-hmm.
So you're talking about measuring the fish and then knowing the age possibly from the size. But what about other stressors, like maybe the fish are smaller in the area because of lack of nutrition or harsher?
Yeah.
Is that something else that you're?
Yeah, so you can also match it up with external data. So there might be other data sources that you can tap into, like sea surface temperature or something like that in that year, and you can look at what factors, and you can actually do statistical models with that. So we'll put in, there's a lot of data that we collect. So I'm actually down there with dive computers too. But you can all use all these different variables as predictors in a statistical model to say what's going to influence fish length. And it would tell you the most likely factors. Does that answer your question?
Yeah, I was wondering if you were looking at more than just the age, you were considering other factors.
So how do you triangulate that, right? You've got data where you get the size of the fish from your tool, but how do you know it's not lying because the fish, the age, you translate that into an age, and then you look and say, wait a minute, how do you check that your actual age guess is right?
Well, we're doing that off of previous species curves. So like a mangrove snapper has a very long history with their age and length relationship. So you can match it up to what we know.
There's been a recent storm or lack of nutrition or some sort of die-off in their food, there might be
like a slower growth.
Offset.
Yeah.
So how do you factor that in?
Yeah. I don't know. I would have to think about that. But there's actually equations that when you put in your length, it would calculate the age for you.
Right.
But yeah, but it might be like a slower growing fish, you mean, but it's still of its age.
Say the population was exposed to some sort of unusual influence that you hadn't accounted for in your data set, right?
Yeah.
And it's this particular, I think is where this question is coming from, right? It's the mangrove snapper eat certain things and those things don't show up that year. So then they're stunted.
Yeah.
And but they're still older.
Yeah.
But they haven't grown.
Yeah.
Right? What do you do about that?
Yeah, it's possible. We would have to look into the literature for how to address that. Yeah.
You'd have to destroy the sample. You'd have to go look at their ear.
To be for sure, yeah.
Sure,
so we
right?
know that you're never going to be
So you have to
exact
grab a
for it.
sample from that population and then back check it.
Yeah. But there's also we also do estimations between, I'm getting into otolith stuff on you, but you can actually look at the size, the space between otolith rings. And people have published that data.
Yeah.
No, internally on the bone. And you can see the space
stuff
between it. Yeah. So you can see if there's higher or slower growth rates. So there is published data on that.
So you think you'd have to go grab a sample just to verify that particular, there isn't a special stressor that year.
Yeah.
To fact check. You have to triangulate.
Yeah.
Yeah.
Yeah. So, but you can build those into your statistical models too, of what's happened, because we've talked about, now this would take a whole nother AI thing that's not on the table right now, but it's not out of the realm of possibility, but you can also look at the background habitat and what's happening. I started this project in August 2023 and everything was bleached. So there's just white patches everywhere in the background. So also, when I do stereo video, I mark what habitat that fish was over. So you can technically do that with the SMILE camera too. But we're prioritizing length right now. But the data is not going anywhere. We still have it. It's stored. You can go back and still continue to collect other data from the images that can be used.
Okay. And how do we check out cameras?
Yeah.
I mean, and there are certain reefs that we can go to, or we can
You can go to whatever reef you want with those cameras. Yeah. So how to check out cameras is you can get in contact with me, or I think we just have a smile@reef.org email now. It'll still go to me. It'll go to me and or Allie, but she'll get it to me no matter what. So smile@reef.org, our website will have that information from the QR code as well. But you can get in contact with me. You basically can just come into Reef and we should have it all set up and ready to go. But, after dives with me, and I've taken everything apart and taken it all out, we can have a camera that's set up in a Pelican case with everything that you need. And you can take it. And as long as you tell me where it went, I don't care where you go. just tell me where it was so that we can look at the factors and consider where it was and what size fish are in that area. Yes.
All right, so I get that you count pixels to get this, and you know the angle that you're shooting,
Yes.
but there's another variable, and that's the distance between you and the subject.
That's what the
Is that what
laser calculates.
How does that work?
From the coordinates. So they know from the fixed position that laser is in. You can't just like put your own mount in
The
there.
distance between the center
Yeah, they know where it is.
So that where it can go.
Yep. So they've even put it out to like predict where the laser would be. Yeah. We can change out the lasers actually. So that's like not a big deal. But where it's fixed on that camera, and this one, why it doesn't have a battery is because it's currently wiggly. So I have to fix it. But yeah, like as long as those, another one got wiggly today. So I have my work cut out for me. Yeah.
Probably there's variance in that too. And you have to calibrate.
Yeah, we don't care about a few millimeters. So if it's like a few millimeters off on a fish, that's fine. And I can tell you some species are a little bit more variable than others from doing the stereo video. So such is life. That's biology. It's got a lot of variance to it when you're dealing with living things. But that's why we like the burst mode too. So we know if you're getting a value that's off by 1,000 millimeters, something's wrong. And I've tossed out fish like that from the data. I'm like, I'm not even using those because they're all over the place. Yep. Yes, Fred.
What's the plan for scientists and fisheries managers to start using this SMILE data?
So that's part of what we want to talk to them about. So the Fishery Management Council, what is the plan for, I was thinking we still have the mic out, sorry. So what is the plan for end users to get a hold of the data and use the data? So that's part of what we're meeting with them about. Like they have funded the project, they're all aware of the project, but we also want to know from them, like, what do we need to prove to kind of trust this data set, improving the validity of it, and demonstrating its utility to you guys so that you can access it. So, right, like it would all be publicly accessible data, much like the Reef data is, but part of what our next steps are is to like build that place that it's found. And also have a visual representation of the data. So we're not going to like tell people exactly where a black grouper that's 500 millimeters is because we don't want it to get speared. But we're still going to have like visual representation of where these fish are found too. So we have lots of ideas for getting it to that next step. But it would be an upload system for citizen scientists as one option, or maybe it's a combined platform that does that, but then they would be able to download the data.
So do you think this would happen, at least get started in the next year?
We keep writing it into grants. So it's definitely, we're aware that we're like the pilot project has worked. We know that it works. It's just tweaking, we can continue to tweak and improve the tech to make the process easier for us, easier for the post-processing, but also what gets it to the next step to being used. And so that's some of the feedback I'll get next week from the council too, is, how do you want that? What would be best for you guys to tap into that data set? So stay tuned, I guess. Any other questions? Yes.
I'm just curious, do you ever tag them? What I'm getting at is, are they territorial? Do they migrate from reef to reef?
Mm-hmm. Yeah. So the question was if we tag them at all to see if they move between reef to reef and if they stay put. Certain species definitely have a strong site fidelity. the grouper are especially one of them. And are any of you guys familiar with the grouper spotter project? So grouper spotter uses another form of AI as facial recognition to look at the patterns of a grouper. So it's mostly being used on my friend Nassau over here with the Cayman Islands spawning aggregation. Anyway, so when I encounter the grouper at Alligator Reef, for example, on the stereo video, I screenshot those black grouper, and hopefully we can use those in the facial recognition technology so we can say that those fish are there. Again, I'm not going to go out and tell the anglers that they're there, but they're there. I know from watching it enough times that it's the same fish. So definitely some strong sight fidelity. It would be much harder with something like a mangrove snapper to be able to prove that. But yeah, we're not pulling anything out and tagging. But I have done that in my past. In my past science life, I've tagged enough fish. Yep.
Will the system work in freshwater without recalibration?
Ooh, that's a
Different
good
fish,
question.
There'd be different fish. but-
Yeah. I don't know. Do you dive in freshwater? Or would the
No,
system work in
people do,
freshwater?
right.
Yeah.
Yeah, and then, seawater, salinity.
Yeah.
it's different.
They do calibrate it, and they've calibrated it in a pool, but they also do it in salt water. They didn't initially do it in salt water. We asked them to.
Because the refractive index is
yeah.
different.
Probably. Yeah. But yeah, like nobody can just go and pick up a camera right now and use their own camera with a laser. But that's another thing we're hoping to do. So that's part of that partnership with, we have a lot of, plans, and they're in the works, which is kind of cool. So the other part of working with Backscatter to make our own laser is to make our own mount that people can go and put on there. So, but we have to eliminate that calibration process down the road, or you have some other in situ process. Yeah. Yep. So we're trying to make the whole thing simpler.
Here's a nerdy question.
No, it's not. You're right on line with what we're doing. If we're gonna, so yeah, hopefully we'll be able to have a product that people can use and collect it all over the place while we're still, but now we're still kind of here working it out. Yeah. Any other questions?
You're going to add some fish moving forward?
We may. So are we going to add some fish moving forward? We might. Right. And that's another feedback I'm getting from the council. It's like, are you good with this list? So we have 2 partners that we work with all the time on the council, but we're meeting with actual stock assessors next week that would be using the data. So we don't want to make the list too long, because we know that could kind of be task heavy for people, or especially if you're doing a survey. So we like to keep it trim, but yeah. So we might bump somebody off and bring somebody on. Don't know. Yeah. Anything else? Don't be afraid to reach out and come out here and grab a smile camera. Thank you guys so much. You can check it out if you want. Yeah.
A certificate.
Ooh.